Squamish, and Tsleil-Waututh First Nation. So, allows us to introduce ourselves. I'm Faisal Tafrialdi with my colleague, Brando, Dani, and Evans. So, let us introduce you to the implementation of this research, which is P53 tumor suppressor gene. So, P53 tumor suppressor gene is the guardian of the genome, playing a crucial role in regulating cell division by maintaining the integrity of the genetic material. It prevents the growth and formation of the cancer cells. In this research, we're using the closed environmental model. These models allows us to simulate the sequence of the genetic mutation, so it allows us to generate the mutational pathway of the P53 tumor suppressor gene. In this research, we're using the SOM clustering method, which stands for self-organizing matrix. This method offers simplicity and high efficiency in our specific implementation. Next. There are two main aims of this research. The first one is to find and identify the characteristic of the cancer-leading mutation DNA, which is the mutational pathway of the P53 tumor suppressor gene. And the second one is to find the quality of the SOM clustering model to cluster the DNA strand by calculating its silhouette score. Next.
So here I will explain you the main step of our method, which is data gathering and clustering. Next. The first one, we also divided into three steps, which the first is next. The raw data collection. The data was collected from NCPA database of P53 gene. The data was contains of string which it have character of A, B, C, and T. The last one, we save the data onto a text file. Next. The second one, we are generate the mutational pathway, which we simulate the cell division using binary tree data structure and also generative tree algorithm to generate the whole mutational pathway. And each generation, we also randomly choose a DNA mutation and simulation. So we're simulating the mutation that happens and also using a mutation that like mama's mutation that which is 6%. And the generation would stop at generation 14, or if the cancer the cell is already cancerous. Next. The third part is we are trying to find which one of the mutation is actually the most dangerous one, or the mutation that is leading to the cancerous cell, which we are using a depth first search algorithm to generate a pathway to the cancerous cell, and then we try to find which one the cancer leading mutation using Lagrange distance. And all this agent would be collected to a table of DNA strand. Next. Going to the second part, we'll also have clustering, which we also divided into three small steps. Next. The first one is data cleaning and encoding, which basically we just remove the duplicates of the string, and also we remove the outliers based on its length, and we also convert the string into a matrix using counting Tumor algorithm. Next. The correlation analysis, we already have around 1,024 columns of substrates, which we need to reduce it to get the best performance. Hence here we are trying to find using correlation analysis, and we pick 5 highest pair of correlation in this data. Next. And the last one, we're using self-organizing matrix to cluster the data, which basically we initiate a random matrix, and then we try to train the matrix to the whole data and find which of these nodes or these grids are the best data representation of our DNA. Next.
What are the results of these steps, you might ask? So here I'm showing you 3 of the most significant data obtained through our experiment that reflects the development of this project. Next. So as you might know, we found 7 substrains that might happen to be involved in the mutation of P53. And in each substring is composed of 5 letters or 5 base nucleotides. So this table shows the correlation between each substring in pairs. As you can see, the different values here means that there are different correlation between each substring. This data were further processed into clusters to extract the further meaning. Next. So here's the cluster. We found 6 clusters that has its own and distinct unique chain. As you can see here, this data with the verdant will be discussed further using the Euclidean distance to find another implications. Next. So we also mentioned about self-organizing metrics. We need a metric size to perform the self-organizing metrics. So we tried every possible value and found that 1/6 size to be the best in terms of their silhouette score. Here in this table, we can see that the high score is happened to be 1/6. So this whole data and this whole result will be discussed in the discussion session by Evan.
Next. So here we discuss 3 main points of our research. Next. The first one is substrate correlation. As the research shows, there are 7 kinds of substrate, and which has a pretty high correlation, which is the lowest one here is only 0.58. Which is shown that this substrate is correlated to each other and it's influencing each other to be growing together or to be decreasing each other. And then what in biological sense this means? This means that these substrates might be representing the functional of its p53 protein gene, which means that this might be the one that check the mutation of the DNA or the one to check, the one to order the cell to be destruct itself. Next. The second one is cluster separation, which we are trying using Euclidean distance matrix for each cluster, and we find this kind of distance in Euclidean, which the highest one is 32.28 and the lowest one 3.86. We might wonder 3.86 might be a pretty small score, but not to forget, we are a 7-dimensional matrix, which means that this is a pretty good separation value to our model. Next. The third one, we compare our model to the other past research, for example in math class by James et al. 2018. We compared first the time complexity. The time complexity of math class is polynomial, while our is linear. While our data is 70,000 DNA strand, by this polynomial, we would take around 30 days to finish a clustering process, while ours using linear is only takes around 10 minutes to clear it. And the silhouette score. Even though our model is too faster and so fast enough to do the clustering, our model keeps the performance almost roughly the same as the math class clustering, which means that this clustering is comparable. Next. And then the limitation of this research, however, we already understand that the data was gathered based on the mathematical model, which means it's not considering any kind of external parameters such as heat or other kind of specific parameter. And the second one, we might not be representing a real world situation, which means that the real world situation, there might be other diseases that might occur to the mutation, but however, we already did our best to do it. Next. And the last one is the future research. In the future, we could use other kind of clustering methods that might be producing a higher performance of clustering. And the second one, we might choose a better parameter for the SLM model, which means its hyperparameters such as learning rate or maximum iteration. Next.
Conclusion. There are several characteristics of cancer-leading mutation that might shift its mutation pair into a cancerous p53 mutation. It was also found there are 7 DNA substrates that might be involved in the functional mechanism of our p53 protein, which its mutation might be dangerous for our cell cycle. The use of SOM or self-organizing matrix as a clustering technique in this unsupervised learning holds promises due to its ability to discover meaningful pattern, correlation, and structures in our data. XLM can effectively organize our DNA sequence into groups based on their similarities, provide valuable insight into genetic relationship and potential functional implication. This reliable approach can contribute to our advancing genetic research and understanding complex biological system. Thank you for your time. Do you have any question?
Next.
Okay, we have 5 minutes for the questions for the audience for this group. Please raise your hand if you would like to ask questions.
Yeah, it seems like you always use digital data for your research. Like, I just want to ask, like, is it possible if we use like the actual data from the cell itself?
Yeah, it's practically very possible to use. Like, for example, we are really using to take the DNA from a cell or doing something like scripting the DNA from its cell and its mitosis to the wet lab. But however, in this kind of research, we're trying to find the characteristic, and then is it a bit hard and a bit impossible for us to doing in this timeline? Thank you very much.
On to add into that, So, and also in past research, the wet lab experiment also holding place. So it's actually possible to use a wet lab experiment to implementation in this research. Thank you.
Mm.
Thank you. Is there any question? Any other question?
It's 4 minutes left for Q&A. Are you guys questions for them?
Sure. So you showed these strings with the letters, right?
Yeah.
And I think that's how the database you worked on, that's what is shown. That is the data, right? A lot of strings with letters. What do they stand for in the real world? How are they connected to the DNA?
So it's basically a base nucleotide of the DNA, which means that are several like A, T, C, and T base nucleotides, which means that we try to represent our base. And the data, as we mentioned, is from NCBI database, which is pretty reliable.
So yeah, so for everyone, these are all groups, stand for molecular groups that the DNA consists of. And so the sequence, when you go down the DNA, is in this form, right? And in biology and biological research, the form or what things look like, they actually are related to function. So even if the letters are the same, the order matters.
Yeah.
And so the research tried to figure out on which so if you disrupt the order, you get mutations, and that can be bad.
Okay. Is anyone wanna else?
How do you find this gene sequence and why this is related to cancer? I think you need a linear plot to show that this the appeal of this kind of gene is related to cancer.
So let me answer that. So our data sequence consists of like the strings of the base nucleotides, right? So there is called p53 protein, which is the guardian of the genome. It makes sure that the transcription of DNA sequence correct, and if the order of our base nucleotide is not in the correct order, it codes cell to die. So in our unsupervised learning method, we are using the mutated p53 model, which consists some mutational pathway.
Can I add into that? So you're asking us how to generate the data, right?
No, I mean, I think there should be a plot or table to show that if the appear of this kind of DNA appears and it lead to cancer, like a plot.
Oh,
and-
yeah. So we're using the like binary tree,
Okay.
so that if there is a cancerous leading mutation, so we already knew that in the end there's p53 tumor suppressor gene that is cancerous. So the mutational pathway, we only using the cancerous p53 mutational pathway. So if there's a leading mutation, we trying to find the characteristic of those leading mutation. So in those one sequence of strand, we need to try to find the characteristic of this one sequence.
I'd like to add one more thing. So it is an interesting question. How do we find the cancerous? So we're using Lafrenstein design. So it's an algorithm to compare the similarity between strands. So we sort it out in one single route, and we find which one is like the one that has like the biggest anomaly, and then we track it back. So that's all.
Okay, I think the time is